Welcome everybody to today's seminar. Our speaker today is Elif Çeltik. It's a PhD plan, so just to remember ourselves, we will have like 20 minutes talk followed by 10 minutes of questions from the audience. And yeah, I think you will introduce yourself, and your talk today will be about the origin and impact of chromosomal inversions on the evolution and physiology of common quails. The stage is yours.
Good afternoon, everyone. My name is Elif Çeltik, and I am pursuing my PhD here under the supervision of Carles Vila and Ines Sanchez Donoso. My thesis is about the origin and impact of chromosomal inversions and the evolution and physiology of the common quails. To briefly present my academic timeline, I did my bachelor about the genetics and bioengineering field in Bilgi University at Turkey. And during my bachelor, I did 2 internships. One of them was about was at Max Planck Institute of Evolutionary Biology, and it was about de novo gene evolution of genes. And other one was at Middle Eastern Technical University, and it was about scopes of migration. And then I did my master in Koç University at Turkey, and my thesis focused on the phylogeny of brown bear populations in Turkey. And since April 2024, I am doing my PhD here, and my study species is common quail. And what is common quail? Common quail is a migratory galliform distributed across a large geographic range, and it's a highly mobile species, and they can breed in different regions within the same breeding season. That means they can breed in the southern Mediterranean areas, and then they can migrate to the northern areas and breed again. That means siblings can born in different regions, and by considering their repeated breeding ability and highly mobility, we don't expect to see any population structure normally. But we found something strange in Iberian Peninsula. In some of the distribution of the population, they have huge chromosomal inversion. And what is chromosomal inversion? That means the segment of the chromosome inverted from its original position, and this can interrupt the recombination between interrupt sequences, and this can lead to formation of supergenes because they cannot recombinate and they are segregating as a distinct units. And with time, these genomic differences can accumulate between inverted and non-inverted population, and this can lead to diversification, even to speciation. And chromosomal inversion are also linked with the behavioral traits and morphological traits. And as you see, I put 2 different examples from birds, and this chromosomal inversion have a huge impact on their phenotypes. And there is a similar situation for our study species. As I said, we have big chromosomal inversion in chromosome 1, and it affects 12% of the genome. And that means this is more than 1,200 genes. And it also affects their phenotypes. And we have 3 different phenotypes. Some populations, some quails doesn't have any inversion. Some of them have inversion in only 1 copy of the chromosome, and we are called them as heterozygotes. And others have inversion in both copies of the same chromosome. And interestingly, this inversion is only found in southern Spain, Morocco, and Micronesian islands, although they have wide range of distribution. And these 3 different genotypes can be found in same place in same time, and they can breed. Because of that, we also have heterozygotes. As you see, these quails are quite different. and the BB's quail with the inversion, and the quails with the inversion have darker throat pigmentation, larger size, rounded wing, and reduced migration ability that affect their reduced flight efficiency that can affect their migration ability. And my thesis is about to understand this chromosomal inversion mechanism and origin and impacts of this chromosomal inversion. And my first chapter is the SNP panel for rapid genotyping. My other two chapters is addressing the question to understand the impact of this inversion, and they are physiological impact and cost of different migratory strategies and diet preferences of different genotypes of common quails. And the other two chapters are focusing on the understand the origin of this inversion, and they are evolutionary relationship between extinct and extinct species and population structure of African and European populations. My first chapter is SNP panel creation. Why do we want to design this SNP panel? Because normally we are genotyping these samples with GBS or other sequencing technologies, but this is time consuming and also expensive, and we want to genotype them or all samples cost effectively. And after genotyping them with this SNP panel, we want to check the distribution of this chromosomal inversion, and we want to use these genotype samples for the physiological analysis, also phenotypic analysis to associate traits with their genotypes. No. Yes, no. Okay. And I will design this SNP panel to detect chromosome 1 inversion, and then I design some primers to detect other inversion, because our colleague found another inversion in chromosome 1, another 2 inversion. Also, I will design this SNP panel for the detect the hybrids. This is not related with my thesis, but this is also something interesting, because we have hybrids. They are admixed with the Japanese quails. Because of the hunting purposes, they are releasing some quails from the farms, and these quails are mostly hybrids or Japanese quails. And we already found some hybrid quails in Donyana, and we want to detect farm origin quails by using this SNP panel. And what is my progress? I already designed these primers, and as a next step, I will test these primers in lab if they are targeting the correct region or not. And my chapter 2 is segregation and senescence of migratory phenotypes. I have two main objectives. One of them to detect the physiological damage associated with migration by comparing quails with different migratory abilities. And other one is to investigate the relationship between fat metabolism and migration using the ghrelin hormone. And as you know, migratory is an energetically expensive process for the animals, and it has some physiological damage. And I want to detect this damage by using different markers. One of them is glucocorticosteroids, especially corticosterone. This is an indicator of stress. I will also measure markers of oxidative stress, and it will show us the cellular damage. And I also want to measure telomere length to understand their cellular aging. And we have a collaborator for this part from the Vienna. She is Valeria Marasco, and she is expert on physiological mechanisms. And my study design, we have 3 genotypes, and we want to check this inversion has any impact on their migration, and to check this physiological cost. Because of that, we will compare these 3 genotypes Also, we will check different age groups and different sex. And other part is related with ghrelin hormone. Our collaborator found this ghrelin hormone reflected changes in fat stores in birds, and it can fluctuate during the migration. Because of that, we captured some quails during the migration season in Doñana, and I want to check their fat store and ghrelin hormone concentration by comparing these quails with inversion and without inversion. And for this part, we already did a lot of sampling. During the last 2 years, we collected more than 170 samples, and we still continue our field work now. And main aim is to collect minimum 20 samples from each category, including sex, genotypes, and age groups. And the important thing during the sampling, we are trying to take the blood samples less than 3 minutes to minimize bias in stress hormones, because as we are capturing them, they are also releasing stress hormones, and we want to eliminate this bias. And then we are directly extracting the plasma in the field and take to the separate tube, and we are freezing them in the field with liquid nitrogen. And what is my progress for this part? We are still continue to the sample collection, and then we will genotype them. Also, we are talking with our collaborator, and we will do a test experiment soon to measure corticosterone and oxidative stress levels. And also, I am planning to do research stay in Vienna for interpretation of my results. And chapter 3 is related with the diet of these quails. These quails have diet including weeds, worms, seeds, and insects. And our colleague Celia Vinagre from our group found something interesting. According to her result, she did isotope analysis for carbon composition of these quails, and these all quails captured in Doñana in same time, in same season. But interestingly, AA quails in wintering time has difference when you compare with the other genotypes, and that means they can have different diet behavior. And to check this further, I want to use DNA metabarcoding method and try to understand if this chromosomal inversion have any impact in their dietary preferences. And for that, I will try to extract DNA from feces. Then I will use some primers to detect plants and invertebrates, and I will identify their diet and try to find an answer if they have different diets or not. And for this part, I applied to the funding from this Society for the Study of Evolution, and we are waiting for the result. And also we are already collecting the fecal samples in our each field work. And then my chapter 4 is evolutionary relationship between extant and extinct species. This part is more related with the find the inversion origin of this inversion. And our another collaborator, Juan Rando, did one study on islands, and in this Macaronesian island he described some species by using bone samples and analyzing them morphologically. And now there are 4 described extinct species from islands. And these all species were extinct because of the human arrivals. And my objective to construct phylogenetic trees between extant and extinct species to understand relationship and how much close these species are. And also I want to check the origin of inversion region by by thinking as a maybe this inversion came from this extinct species with ancient integration. And for that, ancient DNA extraction from bones by already done by Jennifer Leonard and Anna Cornelius. And for these three species, they all had enough DNA, but for Coturnix gomere, we couldn't obtain enough DNA. Then they sequenced these whole genomes of quails, and now I am trying to analyze this data and construct some phylogenetic trees. And I already did one tree by using complete mitochondrial genome, and this is a preliminary, but these are modern common quails and these are ancient quails, and Juan Rando described them differently as morphologically, but according to this mitochondria tree, they look like modern common quails are different from ancient quails. And also, all ancient quails form a monophyletic group. Normally, he described different species for each archipelagos, but they didn't form monophyletic clades, they were all together. Also, Coturnix japonica were more closely related with ancient quails more than common quail. And my progress for this part, lab work was already done, and I am trying to analyze this data. And as a next step, I also want to do phylogenetic tree using the nuclear data. I also want to analyze this inversion region of these ancient DNA genomes. And my chapter 5 is population structure of African and European populations of common quails. As I said in the beginnings, individual can breed in different latitudes and siblings can born in different regions. And there is no differentiation between population from Northern Africa to Mongolia. They are all same subspecies of Coturnix. But when you look at the Africa mainland, in Southern Africa mainland, there are two subspecies. And these two subspecies are isolated populations and they have inverse migratory patterns. Because of that, I also want to check their genomes to check the hypothesis. Maybe this inversion was originated from these isolated populations. And for this part, I started to collaborate with the museum. I already request sample from this museum, and American Museum of Natural History sent some samples to us from Africa, and I am still in the process of finding these samples. And thank you for listening, and I am happy to hear your questions.
Thank you very much, Elif. Now we have 10 minutes for questions.
Thanks so much. That's a beautiful project, and you have a lot to do. I was wondering, so do you know how you're going to be measuring the hormones? Do you have commercial kits for them, or
yes.
Are you developing an assay?
Yes, for corticosterone, we already purchased commercial kit, and we will use this kit to measure the corticosterone level. For other parts, also our collaborator is working about, for example, for telomere, she's working about that, and after we will work with her and decide the best method to.
Right.
But there are some kits.
But for the other hormone that you were going to measure, do you have commercial kits for that one?
For oxidative stress?
No. It's ghrelin.
Ghrelin. For ghrelin, as I know, there is no commercial kits. Valeria Marasco is working about this hormone and trying to find the developed the method to measure this hormone.
If I can explain just very briefly,
Yeah.
that method was developed in Japan, and the team that developed is out of business. So they are trying to import that protocol into Vienna, so they are working hard in trying to optimize, but it's not commercial at all.
Hi, thank you for a lovely project. Maybe a bit of a naive question, but I'm just interested on about the chromosomal inversions. How common are they across the tree of life, and is there something special about quails? These might be very naive questions of why they occur. Is there something special about quails, and how common are they?
I think they are common. As I gave two examples here, but there are also exist in plants and different bird species. I think it's now as I know, it's a bit new mechanism, and people are working on that now. But yeah, when I searched for the articles, I saw a lot of inversion from different species.
Thank you so much for the talk. I also have a question about the physiological approach. So I think you said that you were planning to use individuals with less than a year and then older ones.
Yes.
Which kind of like information at the individual level do you think that you will have? Because so you are using some markers like termers that they are supposed to be aging markers. So can you control for the age of the individual or something like that?
Yeah, we want to use juveniles less than 1 year and more use adults more than 1 year as a control, but also we want to check telomeres because of the migration. Maybe they are their cellular aging is more fast. Then we will use different age group as a control, but our main aim to check maybe because of the migration their aging is different. I don't know if I gave answer.
Thank you. Do you have data on changes in the relative frequencies of each phenotype so that you can use them as a surrogate of fitness?
Could you repeat your question?
Do you have data on how the relative frequencies of the three phenotypes changes over time, over years, so that you can use that information to infer which one has higher fitness than the other?
As I know, we don't have this data.
Thank you for the talk, lovely project.
Thank you.
So on one hand, you were saying that the inversion is associated to reduced migratory capacity, is that the thing? But on the other hand, you have a quite distributed inversion pattern, right? In the Canary Islands, Africa, Europe. So how can you explain this reduced migratory capacity? Because I would assume that this dispersal capacity would be linked also to the migratory capacity. So how can you explain that the inversion is so widespread?
It's not to
or can it evolve kind of independently, these inversions? That would be kind of an alternative.
These species are highly mobile, and they when we check their wide distribution, I think it's a small area has inversion, but I couldn't. Could you again ask?
I'm just thinking off the top of my head, so I don't have any idea. But I would, because if you have this inversion as a widespread pattern in the Canary Islands, Africa, Iberia, I would assume that this inversion could somehow disperse.
Yeah, if they are not flight.
Or the alternative hypothesis,
I think they are still.
would be this inversion would arise independently in the Canary Islands, in Africa, in the Iberia. I'm not sure if this is possible or not. I'm just wondering.
When our group checked their isotopes, they realized differences in their migration, but these quails with inversion still migrate, but I think they are less migrate. I think because of that it's possible to have this distribution for them.
Sure.
Sorry, as requested before, we have a frequency of adults and yearlings for the 3 karyotypes. We did sampling from 2021 until 2023, and they move a lot, and in the same season you can have adults and yearlings, so it's not something that you see from one year to the next one. But what we found so far is that the adults, the heterokaryotypes, we find lots of them. But for the yearlings, we find like way less than is expected. And it's something that we are trying to figure it out why.
Do we have any more questions or questions online? Okay. Then, thank you so much again, Elif.
Thank you.
And thank you for listening, and see you next week with the next seminar.